did a lot of marketing, so now I need to to justify the marketing. So it's a pleasure to be here. This is my second time in Montreal. I've been here in, I think it's end of September, beginning of October in 2019. So after this, we have COVID and whatever. So now I have time to come back again, and so Bahis hosted us for some days. And I prepared some slides, and I will show some some activities that I'm doing and Renata is doing. And the idea is to not, there is, I think, in these interactions between academia and the industry, there is no rules, there is no recipe. You need to test every time, every country, every contact with someone from company, it's different. And we prepared some ideas just to show you how we are working, start projects, and how we can move to interactions with companies. To start, I'd just like to introduce my institution. I'm from São Paulo State University. São Paulo State University, it's one of the 3 state universities that we have in São Paulo. And different from the majority of universities, all these places that it's labeled, it's a city. And in these cities, we have a campus from UNESCO. So this means that it's a state university spread to the São Paulo state. Usually the careers, it's related to the nature of the region. For example, I'm here in Sorocaba. Sorocaba, it's São Paulo city is here. It's 8 kilometers far. And in my campus, we have environmental engineering and mechatronical engineering as undergrad courses, and we have grad courses in environmental sciences, material sciences, electrical engineering, and civil and environmental engineering. This is the grad courses, but this is the numbers from the university. We have this is the number of undergrad courses, this is the number of students, this is the number of grad courses, PhD programs. This is the number of the staff, the whole staff of the university. My campus, it's a small campus. It's quite new. We have 20 years. The university is celebrating next year 50 years, so it's a young university. So this is some perspectives from UNESCO. And also Renata, she works in a non-profit university in Sorocaba. This is the numbers from the university. This is a photo from the campus. The campus is very big and very beautiful. They have more than 6 undergrad courses. This is the number of students. They have master graduate programs. And I worked before in this university for 7 years. And after I moved to São Paulo State University. And I'm there for 18 years now. About the programs or the projects that you get involved, I'm connected with this center. I'm not the director of this center, but I'm the innovation director of this center. So it's expected that I'm the person responsible to get connected between projects, between companies, and to license technology, to helping license technology and whatever. And this is a center that it's sponsored by São Paulo Research Foundation. It's a huge program. We have more than 100 principal investigators involved in this program. You can scan the QR codes to get more details about the center. And in these centers, we have different work packs. We have the synthesis and big data, work pack, natural history, dimensions of biodiversity, microbiome for sustainable solutions. I'm connected here. This center, it's when we are sponsored by this center, you are obliged to have a dissemination. It's expected that you work with dissemination of the results. And they also have a innovation work pack. So this is a kind of program that is sponsored for 5 years, but you can get an extra term for more 5 years. And in 10 years, they expand about, in reais, it's 80 million reais. That it's from Brazilian standard, it's a lot of money for a grant. In this grant, we have scholarships, grants for students, mobility, and whatever. And the idea is that you can understand the biodiversity, behaviors, and you can move until the contact with the society. So the way that you can transfer the knowledge, it's the dissemination, training students, training kids, training the society for the importance of the biodiversity and climate change. And also we can transfer the knowledge through innovation. And especially as we are connected, me and Renata, we are connected with this work packet, microbiome and innovation. The idea is that we can identify some solutions from biodiversity to move to the market, especially to the agriculture applications. And what we expected is that we can make solutions for agriculture get more resilient to climate changes. I mean, like you can develop systems that make plants more resistant to drought, salinity, or whatever, things like this. And in the work pack of innovation, how we are working? What is the strategy that we are working? We are working with the qualification. We need to train students. We need to train undergrad and grad students to be entrepreneur, to have some abilities to be entrepreneur. We needed to find opportunities like connections with environmental innovation spaces. We are connecting to some technological parks at different cities in São Paulo state. We'd like to incubate some startups, and also we are making some internationalization. We are trying to get connections with some different partners to move the knowledge or to transfer the knowledge to another places. It's not only solutions for regional problems or only for Brazil. The idea is that you can get these innovations spread to worldwide. Also, one thing that it's important, it's that we started working with problems and ideas, and here we are stressing the process, because when we have problems and idea, we need to validate this with the partners. I mean, like, oh, someone is facing some problem in on the field. I need to talk to this person and listen to them. Oh, what you are facing? What is the problem? Or why your solution is not solving the problem? So you need to talk to these people, and they will tell you, oh, this is important because there is no way to kill this pest. I was talking this morning with some students, and I remember some years ago, I think 5-6 years ago, I'm attending a program. It's a mentoring program, innovation mentoring program, and I'm part of this training. And we need to validate one technology. And it's a technology for tomatoes. And the person that it's our mentor, our group mentor, she told, look, you need to validate. Go to the field and talk to people. And I'm very happy because I have a very nice solution. I moved to the field, start talking to the guys, the producers. They have very nice places to grow tomatoes. And I start, oh, we have these solutions, and this is because you have this problem. No, I don't have this problem. I say, oh, no, I don't have this problem. I have this problem. This problem I can't solve. And they say, okay, I think that I have some good opportunity, but the opportunity is there, because he told me that this is not a problem. I can control with the products that I have on the market, but this one I can't. There is no solution. So when you talk to people like this, you understand the real problem. So when you are talking about problems, ideas in these projects, we are trying to validate this with companies, because company has problems with the products, because the product needs to increase the performance sometimes. And also we need to talk to people that it's really feeling the problems. And also we have some instructions to how, when you need to patent something, we need to give instructions to researchers like, oh, if you think that you have a good solution, don't publish. Don't go to meetings and publish this, because if you show one poster in some place, that's all. You can't patent, you can't protect, because you publish. You give public the information. So you need to give a lot of instructions to people. And also, as this research center, it's hosted at São Paulo State University. The headquarters of the center is São Paulo State University. We have innovation agents, and the innovation agents help us in contact with companies and contracts and all this stuff. Another center, and this I am the person responsible, I'm the coordinator of this center. It's a federal center. This is a National Institute in Science and Technology. It's sponsored by the federal government. We have a lot of national institutes of science and technology in Brazil, more than 150, in different subjects. So this means that this is an institute that we are interested in development of nanotechnology for sustainable agriculture. This is the only one because they never sponsor two centers in the same or two institutes in the same topic. And this is something that it's provided by, financed by the government. What about the structure of this center? When you create the center, the call where you submitted the proposal, they require us to have a network between researchers, companies, and whatever in different states. We can't have one like a São Paulo state group. No, we need to connect it with different states in Brazil. And we have now 35 principal investigators in this project from different regions. We have people from South, São Paulo, Paraná, Distrito Federal, that is where is the capital from Brazil, Brasilia. This is Mato Grosso. So this is the institutions that it's get involved. And in this project, we have chemists, agronomists, biologists, ecotoxicologists. We have people from agronomists, people, biotechnologists, engineering, a lot of expertises. And we are working together in different projects. It's not everybody involved in the same project, but everybody has your own research line, and we start getting connections. What is the nice point is, as Brazil is huge and you have different biomes in Brazil, we have a good representative areas from biomes that it's involved with agriculture. For example, we have a kind of agriculture in the south that it's the coldest state in Brazil. It's different from the Mato Grosso that it's nearby the Amazonas, and the region is totally different. The biome is totally different. So we can make some solutions, and you can scan these solutions in different biomes, because okay, I can test this in São Paulo, it works very well, but when I move to the south, it's cold. When I move to the center east, it's a different soil, it's different kind of climate. And this is the work packs that we have. So I have people that works with the development of materials, sensors, and as it's a research project, we are trying to understand the interactions of materials with the target and non-target organisms. We have some specific work packs to investigate the activity of the systems in real conditions, like in the field. We have people that it's working with toxicity, risk analysis, and you have also one topic that it's innovation. We have a work pack that it's connected to innovation, and you have the same idea. You need to transfer the knowledge to society through innovation, through patents, to co-development with companies. And you have a work pack that is nanoinformatics, that it's more like this data, how to organize the data from everybody from this program. And what is interesting that we are making connections between these networks. So we have the CBioclima, that it's a center for biodiversity and climate changes. We have different topics. And you have this system, this project that we can develop or apply the technology in the field. And you have some interest topics from both programs. So we have a huge number of scientists that are developing research to connect and to move the applications from one side to another. But what is the strategy? How we started to work with the projects in these programs or in these projects? we consider this scale that it's a maturity level technology scale. And basically, when we have a conception, we are here. When we are doing some patents, we stop more or less here or sometimes here. But we can't move more than this at the university. If you don't try to connect it with companies or with startups or someone that can move this scale up, we can just stop here. We can make very nice publications. We can have nice patents. But if you don't license patents, you don't move this scale. But if you don't start thinking in this scale at the beginning of the program or the project, we can't move this at the end. And why? Because we are not thinking that this can be a new technology. And in this way, we need to validate the projects before we start. Like, okay, someone told that you have a very huge problem with whitefly in Brazil. Okay, this is true or not? It's someone. Okay, I can go to papers and can see a lot of paper talking about the problems with whitefly. But I need to ask the correct person. I need to go to field, I need to go to companies, and they say, no, we have products that we can control. We can't control other things. For example, in citrus, in Brazil, we have a huge problem that it's greening. So this can devastate all trees, orange trees in Brazil. And because of this, the São Paulo State government creates a new center just to study the greening problem, because this is really a huge problem. And what is the main problem? Because one mosquito, they transferred a virus that, or a fungi, I think it's a fungi. They spread this they transfer this to the orange trees, and when they transfer, there is no way to control, there is no solution to control greening. So we are spending a lot of money to try to develop new technologies for this problem in citrus. So if you needed to look at some real problems, let's validate it. We can develop something to citrus. We can develop something to greening. No, it's not our area. Okay, but we can validate it for whitefly. Oh yeah, we have this problem. Yes or no, and you can start checking this. It's difficult to check. Sometimes you need to validate with the agronomists. Luckily, you have people that it's from the agronomy, in these projects, and they are in contact with the producers every day, and this helps us. So we started these things validating the projects. I will show some results. This is some Renata's group results. Renata is specialized in identify microorganisms, and with these microorganisms, she develops nanoparticles, biogenic nanoparticles. And specifically, she grows like fungi, like a trichoderma, and this medium where you grow these organisms, we can use this as a medium to prepare these nanoparticles. And when you prepare these nanoparticles, you can have a particle core and you have like a capping. And in this capping, you can have metabolites, you can find some enzymes, you can find some proteins, peptides, or whatever. And this makes these particles with some potential applications. This is not an organism, this is only a nanoparticles with some capping. And you can use this, for example, trichoderma, it's very used to control this, white molds. And when you use trichoderma, you can control. But you can use also biogenic nanoparticles. In this case, it's iron oxide nanoparticles. And you can get a good control. Because you have some important molecules that can help in the control of this disease. So she is using the microorganisms from the biodiversity to make some nanoparticles and to check the application in some control of some disease. But also she can use the same idea with different organisms, like this is a biological control organism, it's a Boveria, and she can prepare nanoparticles to control not only disease, she can use to control pests. And the idea is the same. Oh, we use a lot of Bavaria as biological control to pests. So she can develop different kinds of particles. She can use silver, iron, titanium, in different materials, silica, whatever. And you can test this in different caterpillars to check, oh, it's better than the control or the organism or not. What is the advantage? These particles can take active for longer. The organism you need to protect. Sometimes you need to guarantee some good conditions to avoid some loss of activity, but the particles can take working for a year. So this is another kind of application. But the idea is, okay, we have these systems, we are using these systems to control pests and diseases. Sometimes it's not working, and why it's not working? Because when you spray some organism in the field, if you have sun, you have UV, and you need to have a good UV protector from your formulations. If you don't have a good UV protector, your organism dies and there is no effect. So this kind of strategy, you can use the organism, some metabolites, some enzymes, some proteins. You can use a nanoparticle support, but if you have the UV, it's not a huge program. You can still work, and then you can control the pests. And also you can use the same strategy, not only for diseases and pests, you can use for biostimulants, as biostimulants. She can use some biogenic nanoparticles, and she can just make some seed treatments or some pot studies, and she can observe what happens as a stimulant for soybean development. So she's using our organisms to make particles to use this as a biostimulant in agriculture. In my case, I'm more focused on the polymeric and lipid nanoparticles. I'm not based on metallic and iron oxide nanoparticles. I'm connected to the polymeric materials. And what we are doing, during our talks with some companies, they are looking for new materials to prepare particles. And they wanted, because of this microplastic stuff, they want to find new surfactants, they want to find new materials, new polymeric materials. And what we did, we got the lignin kraft. Lignin kraft, it's a byproduct from the paper plants, the paper plants. And what we did, we collected this lignin and we start making the fractioning of this lignin with a green solvent. Basically, we use acetic acid, and you make different proportions of acetic acid in water, and you just precipitate different fractions of these lignins. So this is the Kraft lignin, and when you have the fractions, this is the 30% of acetic acid, 40, 50%. You can see that these materials, it's totally different from this one. So this is the byproduct from the paper companies. But when you fraction it, you get materials with different characteristics. And we identify like, oh, what is the content of phenolic groups here? What's the size of the fractions? So we make a strong characterization of these materials. And after we start checking, okay, maybe the fractions that we have a lot of phenolic groups, we can use these as a sun protector in some biological formulation. So we are testing some of these as a sun protector for commercial formulations. Some of these we can use as a stabilizer of nanoparticles. We exchange one surfactant, a classical surfactant, to one of these fractions, and you can get the same particles stable. So we are exchanging, we are offering some opportunities to replace some items from a commercial formulation just using some structures to fraction. This is a feasible process. Yes, it's very feasible because it's like this. We have insoluble, we just start solubilizing in some fractions, and at the end you have some residue, that is this one, and you have different fractions. And the yield of these fractions is high. Each fraction is high. And because of this we check it. There is someone that protected this? No. We make a patent and you deposit a patent and you protect the process. Now what we are doing this with different materials. We are doing this with sugarcane bagasse. We are doing this with we have some coffee peels. We are using this to extract some lignins and fractioning. And now we are getting different materials or different properties with different opportunities. So this is something, it's a case, because we validated this with companies that they need some new materials as substitute to microplastics or polymeric materials. Another thing, another example, this is some science examples. This is something that we validated also with some colleagues. The idea is to develop some systems to release semiochemicals in the fields, because when you have semiochemicals, we can increase the connection between different organisms. In this project, we used methyl salicylate, that well-known molecule that it's released for the plants to attract the natural enemies against pests. So what we did, we developed some prototypes with these nanoparticles containing methyl salicylate, and we check the release profiles, and we put this in contact with organisms to see if we can have some applications. And why we did this? Because the classical way to use the semiochemicals in the field, you have a silicone rubber like this size. You just inject the semiochemicals and you just drop this in the field. And nobody wants to get these silicones that take ever in the fields in the soil. So we are preparing this with some polymeric materials that when you put this in the soil, 2-3 weeks, they disappeared totally. But we needed to have this active ingredient released for 10 days. So that is perfect. We need to get the same benefits, but we are changing the materials. But you need to validate, and this is only a move to show the printer. It's very, very easy. They have a bio-ink. We sure that we developed this bio-ink. We check what is the best material composition or whatever. But it's very fast and very easy to prepare this prototype. And the nice is that when we prepare this, we can more or less standardize the process, because if you do this on the hands, we can't make some lots with the same characteristics. Another example, it's a system that we are using to encapsulate some herbicides. And in this case, we develop a particle that it's made by a protein, a corn protein. This corn protein has low value. There is no huge amount of essential amino acids. So this means that it's from the point of view of food, it's not good. And we can use this protein to encapsulate one herbicide. And what we observed in the systems is that when we spray on the fields, we can decrease 10 times the concentration of the active ingredient when we spray this foliar. We use 2 kilos of atrazine per hectare. This is the standard amount that it's used in the field. And in this case, if you are doing an application, foliar application, you can decrease 10 times. You can spray 200 grams per hectare. But atrazine, it's a pre-herbicide, a pre-emergence herbicide. So this means that I can just spray in the soil and they can avoid that the weeds grow. And when we did this, we can decrease 8 times the concentration. This means that I can spray 25 grams per hectare. So it's a system, because of these systems we are encapsulating, we can drive the active ingredient in a better way to the plants, especially to the weeds. This is a patent also that we have. We protected this before. And okay, you have this case, but what we can do this? Oh, always we protect and we publish. We make this public because people can get access. But what we started doing? We can stop and we can move to a new project? No, we need to find someone to move this forward. And what is the idea? We come back the same. And now we start increasing the TRL. What is the way? We can license the patents, so we can find companies to make the license. We can make co-developments with the companies. The company comes and say, oh, I'm not interested in atrazine. I'm interested in a new herbicide. Okay, we don't have this, but we can develop a new system with this, your herbicide, and you can make a project with a company to develop a new herbicide. It's a different herbicide, it's a different nanoparticles, a different topic, but you can do this. And also, with these companies, we can move to the field. We can do real condition validation, because sometimes in the lab, we just make some pots, we can use some greenhouse studies, but you don't move to the field or to real conditions. So this is the strategy. Now we have this project, we have these results, and how can you reach the companies? One strategy, I was telling someone this morning, we moved to meetings from the agronomists, because where is the agronomists? The companies are there to show their technology to the agronomists. And we started going to these meetings, and we present. Sometimes we have a talk, sometimes we have a poster, and during these talks we can show some of these initiatives, and they say, okay, let's talk and let's see some possibilities to make projects. Just to have an idea, because of this knowledge that Renata got with the biogenic nanoparticle, she opens her startup. It's called the BioBio, and what she's doing, she's looking for different microorganisms. She's going to the biodiversity, identifying different microorganisms, and she's validating these microorganisms as a biological control, as a substrate to make these new nanoparticles to pest control, biostimulating, or disease control. So this is the subject of her startup. She's doing some studies in real conditions with some hortifruti. This is some controls, studies. This is a commercial product, so you can see the roots here. You can see the roots here, and when she uses her product, you can see the difference in the root system. So this technology, it's important to increase the development of the roots. If you develop better the roots, you get more nutrients, and the plants get more, the quality it's increasing. She's validating this in different conditions. This is look at the size of this farm. This is a green bell pepper. This is the farm standard. And this is also interesting, because all farms, they have their protocols to control everything, or to protocols of nutrition and pest control. And you just change, or you just add your products in this farm standard protocol, and you check what happens. Especially here, the photos, it's not so good, but basically you can see that the quality of the vigor of the plants, it's different. If you have the vigor different, it seems that the plants, it's growing. And also she's validating this in tomatoes. So this is the plants treated with her product, and this is the plants treated with the commercial product. So we are validated with someone that has some products on the market. In my case also, what is the way that I started making interactions with companies? As I'm a public employee, I can open my own company, but I can't have some official position. I mean, I can't be CEO. I can be only part of a company. I can't develop anything. I can't work to the company. So if I have a company and I can't work to the company, I don't think it's a good way. But I start training students or training PhD over the last years. And some moments, some group of former PhDs, they decided to open one startup. The name is Binano. And what is their strategy? Immediately, as they are a company that comes from the university, they have a stamp, like a label, that, oh, you are a daughter company from UNESCO. So you have like a stamp that say, oh, you are a university-based startup. And because you are a university-based startup, you can license patents from the university with no costs. And they just license different patents from the university. And now they are trying to make these patents or this innovation as a product. Just to have an idea, they licensed that patent to atrazine, and they are now look at the difference of the way that they show, because they have a market strategy to show the products or to show that you decrease the amount of active ingredients. They are validating this in real conditions, in field conditions. It's not more important conditions, but they are trying to use some strategy to show the benefits of this. Also, they are using not only these patents, they are developing something. This is a paper that they published in 2019. It's some nanoparticles with some plant growth hormones. And we run a field study in that time with tomatoes, and we increased the productivity of tomatoes a lot. Okay, we just published, but now they are developing different systems based on this publication as a biostimulant with different plant growth hormones. And they are validating this because they can decrease, in the case of gibberellic acid, 2.5 times the concentration of gibberellic acid. Gibberellic acid is a very expensive molecule. So if you have a product that you can decrease and you make the seed treatment, you can try to develop a new product that is cheaper. And they are validating this in field conditions. So this is some corn. This is a farmer. They make an agreement with the farmer and say, oh, can you give us some space? We can just treat the seeds, deliver to you, and you take care of everything. And this is the differences from the corn from the farm, the standard, and this is the corn. At the end of the production, they tested different formulations, different seed treatments, and what they observed that in some moment they increased the 25 packs per hectare. It's a very interesting, because when you increase the number of packs per hectare, you can cover the costs of these new products, and maybe you can get an extra revenue because you are getting more productivity. So they are validating this. And also, it's not limited to this, but these systems that we can decrease like 80 or 10 times concentration, depends on the way that we are spraying. We connected to this company. This is it's a US company. Basically, they have these robots, and in these robots, these robots, it's self-guided robots. They can scan the field during the day, for example, or during the night. They have some solar panels and some batteries. And what they did, they have intelligence artificial models here, and they can recognize what is corn, what is weeds. And what they have, they have this spray parts, wings, and each wing can be opened in different moments. I mean, you are scanning, they open, oh, this is a weed, they open to spray here. But there is no more weeds, they close here. But there is here, they open here. So they are spraying very locally. They are spraying very locally, so this means that you decrease the spray of active ingredients in the field, because the model that we use it to spray in the whole area. And in this case, they are spraying locally. But let's try to spray locally with a formulation that you can decrease 10 times the concentration. So I'm decreasing even more the concentration of active ingredients that I'm putting in the field. So we are validating the technology in the spray process, because you need to validate this. And also we can, this is I recorded this. This is a field trial that we did all together, because we moved to the farm. The company has their own experimental area, and we have the corn and you have the weeds, and you make different treatments. And now we are checking the results, when you can check the results with the productivity of the corn, because if you control better the weeds, there is no too much competition for nutrients. The corn can grow, and you can check the productivity, and that's all. So it's a very nice strategy, and we are connecting different technologies, startups, to validate the products. But we come back here. So we are here. We can increase the TRL. We can validate it. We can start developing, our companies start developing some scale-up process. But when to move from here to real the market, this is the real problem. So this is the moment where the things stop, because now we need to find good companies or big companies that can come here and get their technology, or we can look for funding for these startups to building or to produce this in huge amounts to sale, or they need to make some cooperation with some scale-up facilities. So this is the main issue that we have at this moment. When I say we have, it's because, okay, we are here, and as a researcher, I would like to see something here. Sure that I need some companies here, but it's important. And this is the most expensive part, and you need to find some ways. And this is really a gap that you have at this moment. So to finish, as a conclusion, we needed to get strengthening the connections between university and the innovation and the biodiversity, use of the examples from the biodiversity to be to solving problems on the field. We need to advance in this TRL with the projects, and this strategy, I think it's very nice because we are always thinking in a product. It's important to tell that we are interested in answer basic questions. We are interested in understand the mechanism of interaction of nanoparticles and plants and nanoparticles and organisms. But if we can do this, look into a next step that is to try to transfer this for some companies. We need to look for international collaboration, because sometimes the problems that we have, it's not only from Brazil, it's worldwide. And to change the expertise, it's important. And also we need the challenge is to transfer the knowledge that we are developing at the university to the real society that it's needing some solutions. So this is some conclusions. I'd like just to thank the sponsor agents and the institutions, and here can get our contacts. And I'm open for questions. Thanks.